Thank you, Dr. Tanaki and Antonio, for giving me this opportunity to talk to you. It's a little bit of the work that we're doing and some of the challenges and opportunities presented by it. So we're trying to develop a public genetic diversity research platform where genetic resources serve as a foundation, and whereby undertaking genotyping and phenotyping, we can bridge the genotype to phenotype divide. And by doing that, for addressing current challenges for plant breeding, we'll have a sustainable platform which will also allow us to add value to the gene bank collection, improving its conservation and utilization over time. Underlying this is the need for extensive use of computation, bioinformatics, and databases. So our research at IRRI is organized under the Global Rice Science Partnership, which includes not only IRRI but other institutes working on rice in the CG system, Africa Rice and CIAT, but also Seurat and IRD in France and at other sites across the world, but also JIRCUS in Japan. These are the main founding partners of GRISP, but we also have on the order 950 other partners in advanced research institutes, national agricultural programs, NGOs, etc. So under this framework, the product line 1 involves genetic diversity and its characterization and use. So this talk deals with the product line I'm involved in, characterizing genetic diversity and creating novel gene pools. And under that, we have our RiceNIP consortium, which I talked about in the plenary session on Monday. But we also have the phenotyping network, our sequencing effort, which Jikong Li presented some of the current data yesterday. And we also have an activity to develop new genetic stocks, including RALs, NAM populations, MAGIC populations, BILs, etc. Also in this program is the C4 project, which you've heard a lot about at this conference. But we also have activities to undertake the characterization of high-value genes. So these activities are contributing extensively to the ability to undertake genome-wide association studies. And all of that now we're envisioning as a larger program, perhaps. And we came up with the acronym ASPIRE because it's what we're hoping to achieve, for one. So accelerated sequencing and phenotyping for improved rice. As a foundation for that and for the activities, we have a large range of different sets of germplasm that have been collected, sometimes used in other programs, but also new sets of material coming into it, including just today at IRRI, the Embrapa core collection of 540 lines was received. Brazil is a party to the international treaty, and this is a big step forward for Brazil to begin freely exchanging their germplasm. Currently, it's in excess of 10,000 purified genetic stocks that we have available for different projects, including going beyond the 3K genome sequencing project. And the management of that material and its entry into a database system is under the International Rice Gene Bank Management System. And we cannot distribute the material until we have a record in our database, including the assignment of a new accession number that allows us to track and associate IP with the material. Plus, that happens when we have sufficient stock. So in terms of the purification, we've taken a single seed descent type approach. We're increasing material after it's been purified so that we have enough material for phenotyping. Sometimes it's now approaching 3 kilograms. And then for the genotyping and sequencing, we've taken an additional cycle with a single plant for the DNA and then saved the seed from that individual and that seed is being archived. It's not going into additional increase at this time. The Rice SNP Consortium, I'll just briefly go through this. I talked about the diversity panels, one from USDA and Cornell, and the one that we developed in total for 2,000 lines for genotyping on the high-density arrays. And the arrays, 700,000 SNPs developed at Cornell from a SNP discovery pool from a wide range of collaborators and donors contributing to that. And now we've got genotype data almost complete on the 2,000 lines against that 700,000 SNPs. For the global phenotyping network, we're encompassing as many traits as possible, including yield, disease resistance, blast, brown spot, bacterial blight, fault smut, but also abiotic stresses, salinity, drought, heat, grain quality, seed physiology, etc. Name a trait, we want to collect data on it. This is one example of phenotyping for drought stress on the AUS collection in that 2K panel. It's work by Amelia Henry, funded by the Generation Challenge program, and traits like early vigor, canopy temperature, NDVI, yield, shoot, and root biomass were taken in the field and in this greenhouse under controlled conditions. And then also this was mentioned in the talk earlier, the Global Rice Phenotyping Network, one project under it, Phenotyping Rice for Advanced Yield, or the PRAY project. There's a very wide range of traits that are being phenotyped under that project, including leaf anatomy, and these are recent results from Paul Quick's team and Jackie sitting in the back of the room. And they, when that panel of 295 accessions was grown this past dry season, they went out and they collected leaves, and then they undertook different leaf anatomy measurements, including inner vein distance, this vascular bundle for small veins, for large veins, etc. And for the range of traits then that they measured, and then Jackie looked at correlation, pairwise correlation between the different traits, and some traits were not correlated, such as delta, carbon-13, discrimination, other traits like small vein vascular bundle. No. Hold on. Next slide. So traits that were not correlated were delta for carbon-13 discrimination, leaf thickness at the small vein only showed weak correlations, vein density only showed weak correlations, but vascular bundle total area at the small vein and vascular bundle total area at the large vein were strongly correlated. And this is just a first pass association analysis with the cutoff for minor allele frequency of 5% and eliminating SNP loci with missing data points, so forth, using tassel. And there are a number of peaks showing up that merit following up and further analysis using tassel or MIX. In terms of the sequencing effort, Jikong described this a bit yesterday, but we're working both with BGI Shenzhen and CAS initially to sequence 3,000. The final sequencing data was fixed in about May on the collection. The fundings come from the Green Super Rice project by the Gates Foundation and the Chinese Ministry of Science and Technology. Now that's 3,000 lines fixed at greater than 10x depth with 17 terabases of trimmed reads. Jikong reported that there are 20, now we know that there are 25 million loci in the rice genome relative to Nippon Bari where SNPs were called. Also, he showed the phylogenetic reconstruction that demonstrates that the 5 representative groups we expected to see are there. BGI completed basic analyses. They were pushed to the Chinese supercomputer Tianhe 1, and interim data analysis consumed over 1 petabyte of storage to do that. Over 2,000 of the genomes now are at gigadb.org, which is a companion database to the BGI Biomed Central new publication called Giga Science Journal. And in the GigaDB and in Giga Science Journal, each sequence file can, or a collection of sequences, can get a data object identifier. So it becomes a tractable, a citable micro reference in the literature. And we're following Toronto agreements, so the raw data will become public after embargo. Prior to publication, we're using Galaxy to document workflows and analyses. We want a white paper publication out soon describing the project and its goals, and data availability and pathway for data analysis. And we're also, as he mentioned, calling for a global consortium of partners to extend data analyses and follow the framework of the Human 1,000 Genomes and Code and other projects. So we also are forming a scientific advisory of experts to guide going beyond the primary analysis. So people with expertise in large-scale projects, database development, sequencing technology, etc. And we've also been talking to a number of different groups who have expressed interest in collaborating on this effort, including NIAS, Cornell, TGAC, the Genome Analysis Center in Norwich, Klaus Meyer at MIPS, our GRIS partners, Surad IRD, Chinese Academy of Sciences, CAS, BGI, Academia Sinica, our colleagues in Taiwan, MPI, hopefully Detlef Weigel's group, and Kunming Zhu Institute. So we've also been making specialized populations for genetic studies, and Matthias Loria has been working to create, it's about 20 RIL populations. They're now at F7 bulk stage for a nested association mapping panel. We're at IRRI, we've been developing a collection of populations too. We have 5,000 now F7 bulk lines from 3 populations, and we're advancing about 12,000 RILs from 32 selected crosses. They are at these different generational states now. One of these populations is going into a phenomics experiment with Jan Leach and collaborators called ERISA Phenome 1 to help us move through the phenotyping bottleneck. And in that, it's IR64 crossed by Aswina at F7, and they're segregating, this population is segregating for biomass-related traits. Also shows segregation for flowering time, for panicle weight, as well as biomass. And in that design, we have now a field-based phenomic system at IRRI. There are sensors mounted on a boom that's on a precision-guided tractor that can move very slowly and accurately with GPS, a GPS system that's accurate to 2.5 centimeters placement. And I hope this works. So this is a movie of the boom riding through a trial experiment in the field across plots as just a trial for the big experiment that's going to be done this coming dry season. So Hay sitting in the back loves this movie. So, but for the sake of time. So this large experiment, 1,500 RILs, grouped block design, and massive amounts of data to be collected. So we're really facing an onslaught of data. And that's giving us a number of challenges as well as opportunities. So hopefully we can begin to understand the genotype to phenotype divide, and there is the need to be able to integrate across these different diverse data types, and that other talks have addressed this. So key challenges are how we deal with thousands of genomes and thousands of genotypes. We're looking at storage and archival using bioHDF5, as well as distributed federated type databases to integrate data. And we're definitely following existing examples on the creation of such a system. But also in terms of tools for data mining and linking genotypes to phenotypes, there are tools in progress, some of which are coming from, well, different groups across the world will adopt and adapt as necessary. So one crucial aspect to a federated distributed system is that the databases need to really make use of semantic web and web services, as well as controlled vocabularies and ontologies. And there's now a push, Pankaj Jaiswal in the crop ontology, to get the plant ontologies up to a reference type ontology state. And on doing that, if the plant ontology and trait ontology, environment ontology, etc., get to the state of acceptance as the gene ontology, then the ability to cross-query databases will become very easy. There's also a desire to get the terms and the ontologies better defined so that you can, if you want a trait or some other descriptor, and instead of creating a new definition, draw on the existing definition. So in the Chemi database, there's the term ABA. In the measurement database, there's concentration by HPLC. In the environment ontology, there should be a term for upland water-limited environment. And then the plant ontology leaves at a certain developmental stage. So if you have a trait that you're measuring ABA in the leaves, you can do the cross-reference between these terms and get a precise definition of that as a way to curate the term. And that could also be related to a gene ontology if you have, for example, a microarray experiment, and you see that 9-cis-epoxygenase at the bottom of the long arm of chromosome 12 has been expressed. So, almost. So, at IRRI, we're using the International Rice Information System, and it can link across different types of data, including the Genetic Resource Information System, the Pedigree Management System, but also field data. The Gene Management System is less mature than the other three, as well as Location Data Management System. And this is just a screenshot. If you go to our website, irisiri.org, and then if you search for IR64-21, and this is the accession number corresponding to the genetic stock that was used to create the mapping populations. And then this is the management domain of seed increase for that particular accession. And then if you float the mouse over one of those boxes, then you get a URN descriptor, meaning that this is already web service enabled. So you could link your data in your database to this database if you embed the same type of description in your database. If you have a GID or your own descriptor cross-reference to the GIDs in our database, then you can draw any of the information from our database into your database. So federated, we're also looking at the phenomics ontology-driven database. It's a development out of CSIRO. It is also using ontology as its framework for its schema, and it's using OW and RDFS for the way it expresses ontologies. So we're adopting this for our phenomics data, and Xavier Sol, one of our collaborators, was key to its development. I showed this before. We're also, we have an instance now of the UCSC browser for our SNP and resequencing data that will soon be populated. So for Aspire, global consortium of partners with the scientific advisory panel, raw data made freely available, in the process of adopting or building tools for linking genotype to phenotype. It will be a federated, make use of federated systems, so we're not going to reinvent the wheel. We'll draw on existing resources as much as possible. But to make it sustainable, we're looking at a public-private licensing type mode so that it will be secure in the long run. We're also now, whereas all germplasm was freely available from IRRI, now at least the private companies are going to pay full cost recovery. Bob Ziegler at a forum in Singapore by the Economist coined the term public PPPP, public pays, private profits. So our funding at IRRI is pretty good right now, but it's by no means secure, and I'm sure that is the case for a large number of your institutes. So all of this, all of these activities, the SNP chip, sequencing, phenotyping, stocks, association genetics, then we have the whole gene bank and all of your breeding populations, etc., as a reverse genetic system. But for the phenotypers out there, you have to be prepared to do a lot more phenotyping, because that's now the bottleneck, and we need that data in order to make sense of the genomic and the genotyping. So, thank you.
Thank you, Dr. McNally. So, not only for this presentation, but also for every presentation, I'd like to make some question time. So any question or request for every presentation is welcome. No question? So thank you, McNally.
I think everybody's tired. It's
it's no problem. Maybe you too.